So hello, everyone, and thank you for joining us today for a very special discussion with Dr. William Powell. And we'll be talking today about his book, Breakthrough, which chronicles the incredible journeys of how 8 different drugs made it from idea to medicine. And we'll also be talking about his experiences and perspectives on leadership, particularly when it comes to this very daunting but also very rewarding task of drug development. My name is Jonathan Yu. I'm the Chief Operating Officer of a company called Disc Medicine, and we're a biopharmaceutical startup company focused on developing novel therapies to treat serious and severe hematologic diseases. Before we dig in, I wanted to say a big thank you to Elaine and the Elevate leadership for organizing this discussion. For those of you who may be less familiar, Elevate is a nonprofit focused on advancing Asian American leadership in biopharma. It's an incredible community, and it's a treat to be doing this today. For the audience listening, feel free to share reactions and questions in the chat. Thanks to those of you who've already shared questions in advance. We'll weave those into the discussion today. And we'll also try to keep an eye on what resonates with you and what you'd like to hear more about as well. And lastly, today is February 11th, so happy International Day of Women and Girls in Science. Today we're talking about breakthroughs and all the breakthroughs that have occurred in the past and that are going to come to the future. These simply would not be possible without our colleagues and women colleagues in our industry. So thank you, and we hope there will be greater representation in the future. So with that, let's get to it. So William, thanks for making the time today. Maybe we can start by having you tell us a little bit about yourself. You've had an incredible career path through medicine. Tell us a little bit about what brought you into this field and your path of becoming a physician and the researcher and drug developer and now CEO of the company.
So, Jonathan, first of all, thanks a lot for agreeing to host today. I really appreciate it. And I also want to thank Elaine Chen, as well as the Elevate leadership, just like yourself, for giving us the opportunity here. Um, yeah, I basically, my parents were originally from China. They actually came before 1949, at a time when there were very few Asian Americans in America, actually. And then I was born in DC, grew up in the Maryland area. Both of my parents were doctors, so it was always assumed, I guess, that my sister and I would go into medicine. But it really didn't hit home for me until my dad died when I was age 13, unfortunately, of metastatic colon cancer. It was a little bit unexpected. He had gone through chemotherapy and surgery a couple of years before, and then it had come back, and unfortunately, then went very quickly. So at that time, I did vow that I would try to make a difference for patients like himself. But being age 13, I didn't know what that would really entail. And then basically after college, then went to medical school, did an MD, PhD, internal medicine, medical oncology, postdoc, many, many years of training, like many people on the call here, I'm sure. And then ultimately I ended up doing translational oncology, first starting in academia at Memorial Sloan Kettering, where I was fortunate then to be in the postdoc in the lab of Harold Varmus, and we and others at the time found EGF receptor mutations as a genetic basis for why new drugs at that time, Aressa and Tarceva, only worked in about 10% of patients. So that really whetted my appetite. I did see, for example, many lung cancer patients having a Lazarus-like effect. When you take this pill and literally within a week, they become fully recovered. And really that got me very interested in first understanding, how drugs work in the first place, doing translational research, but then ultimately I helped other companies like AstraZeneca develop molecules and so on, and then I got recruited to pharma. I did have a big, what should I do in 2013 sort of moment when I was in academia. I was already division chief. By that time, I was at Vanderbilt University, was a division chief of HEMONC, still had a lab, saw patients, and helped to run trials. But having helped other companies, I was a real fork in the road. And I did decide to take that fork into pharma and develop drugs full time, mainly because I wanted to be able to have an impact on thousands, if not, ultimately maybe millions of patients across the globe rather than just on a one-on-one basis. And I haven't looked back. Then I was in pharma for about 8 years. And then now I've been in biotech for the last couple of years.
Yeah. No, thank you for that. And I think that's one of the things that, whether you are a physician or in academic research, or even at, in large pharma or in a startup, it begins and ends with a patient. And I think that, the scale at which you can impact patient care, particularly when you're looking at working at these large pharmaceutical companies, I was at J&J for some time. It's really completely rewarding, but also, you can still have a huge effect on a patient's life. Um, so you've been a leader in medicine in different settings. These are all very different organizations. Maybe you can tell us a little bit about, you know, and all with the same kind of mission. So how have you observed leadership to be different in these different settings, whether as a physician or in global pharma, and now in a as a CEO of a startup?
Yeah, no, that's a great question. I think vision always starts with, leadership always starts with a vision. And in medicine, at least, the ultimate vision is to help patients improve their lives, whether you're as a doctor in an academic setting, whether it's through research in an academic setting, or whether it's in developing drugs in a pharma or a biotech. So if you always put the patient first, I think you can't go wrong.
Yeah,
But yeah, I would say from a leadership perspective, it's very, very different. In academia, well, obviously you have to work with a lot of folks in the hospital. So you want to, bring together people in terms of trying to help patients. But when you're in academia, I would say, particularly like as a division chief, what's interesting is you think you have some so-called power, but you don't have very much power. I'll give you an example of that. When I was interviewing with Art Levinson actually for the Roche position, which was initially head of oncology at Roche PRED. He's like, yeah, you think you have power as a division chief, but you really don't because your academic scientists, they depend on their grants, they depend on their own publications. You can tell them what to do, but they don't have to listen to you, and so on and so forth. So I didn't really quite appreciate that until I got to pharma, where yeah, like if I said something, then they had to do it, right? Or there were potential consequences. And then similarly in biotech, it's just very different in terms of leadership. So I think in academia, you can obviously lead a division, you can lead a department, and so on and so forth. But a lot of the academics and the scientists really lead their own labs and their own careers.
yeah,
But in in biotech, you're working much more as a team. People have to really be part of the team in terms of rowing in the same direction, and obviously make milestones and so on and so forth. So it's just a different kind of leadership.
yeah,
I would say in each arena, though, you sort of have to understand what are the metrics for success, and what are the goals that you do want to set, and what would resonate with the different people. You do have to talk to different people in different ways and really make and convey what you want to say on their level. And you can't discount that in terms of a leadership and the effectiveness of communication.
yeah, yeah. No, that's great. Um, I do want to talk about the book, but before we talk about the book, we should talk about your t-shirt. And like
Oh,
right now, the camera's cutting off the bottom half, so it looks like you're saying no research, but I don't think that's your mission here.
there you
So,
go.
yeah, so no research means no cures. And I think we'll be talking about how later on today, like how innovation is really stacked and it really requires you from beginning to end. There's like this handoff. It's both a marathon and a relay for things to become a drug. So, it's important to just keep feeding the pipeline. So, we'll get into more of that later on, but it was important for us to mention that you are not about no research. You're saying no research means no
Yeah, no
cures.
research, no cures. And we'll talk about that as we get into the
Yeah.
book.
So yeah, let's get into the book. How did you come about writing this book and tell us a little bit about, the structure, why you decided to structure it this way, going through these case studies and, what was the motivation, the genesis, and how did you get these great stories?
Yeah, there were several factors that were coming into play basically during COVID. So I used to travel about a third of the time. So I was obviously on planes a lot. But after COVID hit, then I all of a sudden had a third of my time back. I was the head of Roche Pharma Research Early Development at the time. And in March 2020, not only was managing Roche PRED, trying to help with COVID as well, but then we did know that a molecule called risdiplam, which is a oral RNA splicing modifier molecule coming from Roche PRED, was going to get approved later in 2020. And so I wanted to commemorate the so-called heroes of that story. But then I also had an ulterior motive, which was, I wanted to build the most innovative research discovery unit on the planet. And so I thought, well, if I write this story, maybe I'll get some insights as to how this particular team was successful and some of the things they had to overcome and set an example for the other teams
Hmm.
doing drug discovery. In any case, I wrote the chapter, and to write the chapter, I started from the academic discoveries and then went to the biotech, that PTC Therapeutics that, made the initial lead molecules, and then described what actually had to happen in Roche PRED, which included not only, taking the lead compound, but making it to the final candidate, and then the setback that happened in phase one with the first molecule and how the team had to go back to the drawing board, bring a second molecule in, and so on and so forth. And it really resonated with a lot of people. And so then I was like, well, maybe I could write a book about innovation in drug discovery, but I don't want to write it just about Roche molecules because then everyone
Hmm.
think I'm just advertising for Roche. So, still COVID, I took on a whim, basically emailed my colleagues, heads of R&D that I knew, and emailed like Jay Bradner from Novartis or Merdad Persay from Gilead or Susan Galbraith from AstraZeneca, Michael Dolson from Pfizer.
Hmm.
And I basically said, hey, I was thinking about writing this book about innovation in drug discovery, really to highlight, what happens inside pharma in addition to what happens inside biotech and academia. And it has to be a molecule that shows not only cutting edge innovation and high impact on patients, but also illustrate the resilience and persistence that teams have to go through to ultimately succeed. So I really thought that they would say no, given, that we were like frenemies or, friendly
Uh-huh.
competitors.
Uh-huh.
But to my surprise, they actually said yes. I think if they had said no, there would be no book, obviously. But they said yes. And so I ended up then choosing eight different molecules from eight different companies. Seven of the molecules are new, relatively new, even just approved. And only one molecule is old, paracetamol or Tylenol,
Yeah.
coming from the 1950s or so. But yeah, the criteria to be in the book was it had to be really cool science, it had to be impactful on patients, and then third, yeah, it had to illustrate really the challenges that had to be overcome to be ultimately successful.
Yeah, I mean, I think it's so reassuring to hear that these other heads of research were so willing to contribute these stories because I think it tells me that, you know, everybody who's leading research at these large organizations is sort of coming at it from the same spirit and like wanting these stories to be told and, resilience, perseverance, innovation, intellectual curiosity, those are all things that I think are what keep this process of drug development to a certain extent, pure. And so it's great to know that, large pharma is like that. That's what's driving the spirit there.
Yeah, and I
Um,
think the other point I wanted to make was, as I mentioned earlier, was to highlight the unsung heroes of drug development. The academics, like I've been on all the sides, so this is not a criticism or anything. Academics usually get a lot of the credit for the initial discoveries, and there's no discounting, the amazing basic science that is done. But to transfer or to transform an idea and a finding in the basic science laboratory ultimately to a marketable drug that can be given to millions of patients around the globe takes an enormous amount of effort, enormous amount of people, and a lot of trial and error as well. So it should not be, as you know, a given. And so really wanted to highlight those unsung heroes of drug development.
no, absolutely. Yeah. In fact, that's kind of the thing I wanted to talk about next in that, you read all kinds of statistics and much has been written about, the cost, time, risk. I mean, we kind of live it, no matter where you are in the biopharma ecosystem about just how extensive the resources are to get, associated with drug development. But, reading it in this form really made it seem like it's kind of a miracle that any drug ever gets developed. So, yeah, maybe talk a little bit about that. You already alluded to it, but is this something you wanted to communicate and tell us just a little bit about what it takes to get a drug over the finish line?
Yeah, well, so first of all, you have to start with basic science research, which is
Yeah.
why I have this T-shirt. I do, having written the book and also having been from academia, I think all of us probably also trained in academia at some point. The importance of basic science research can't be taken for granted, and we need to continue to invest in that. And so, really do think, that no research, no cures down the line, right? No research today is no cures tomorrow. In any case, yeah, so I think the ultimate challenge is even if you have a great target, if it's a new target, you may not understand its biology for a long time. Even if you do then understand its biology, then you need to understand how to drug it. and you may not have the technology to drug it at that time. And then ultimately, you need to understand which disease that target is relevant in, and then not only which disease, but which state of that disease. And I think all of those elements, the biology, the technology, the clinic, they all have to come together. Sort of, you think of a Venn diagram, and you have to get to the center of that Venn diagram in order to get to a final molecule.
Yeah.
The reason I re also wanted to write the book a little bit was because, at least in big pharma, there's a lot of talk about these R&D productivity equations. All you have to do is reduce cycle time, increase the PTS, and so on and so forth. And, that really doesn't count in the factor of innovation that has to happen. Of course, if you go after a really established target, it's much easier. But if you're going after a completely novel target, you really have to be persistent and resilient and learn as you go.
Yeah.
Yeah.
Exactly. I mean, that was one of the things that struck me about, I think, almost every single one of these stories you were telling, in that it's not just one aha moment. It's just at the handoff of each stage, every part of the value chain, there's opportunity for and almost a need for innovation. Just because about, elucidate the underlying biology, you have to still make the drug. Once you've made the drug, you now have to figure out how to test it. Once you've tested it, you have to figure out how to like manufacture it at scale. And each of these requires some spark and ingenuity. And if at any point anybody dropped the ball, the process stops. And so
Yeah.
it's really remarkable. Um,
And that's why, as you know, sometimes the process can take, 100 years, so to speak, from
yes.
initial clinical description to a final molecule. Now,
Yeah.
the pace of research and development is much, much faster now. So hopefully we're um, doing things much faster. One of the chapters is about Paxlovid, as you know, which was developed in a record 17 months.
Yeah.
And things are getting faster, but it still takes time for all of these things to mature so that you can get to the center of that Venn diagram.
Yeah. Maybe digging a little bit deeper on the notion of resources. I mean, one of the key ingredients to overcoming the odds in developing a drug and breaking through is really being able to get the resources for it. You have a chapter, and you alluded to this earlier, about the EGFR inhibitors. It actually spans multiple decades in the biology to elucidate growth factors and all of that. But the development of this class for lung cancer that culminates in the approval of Tagrisso, a third generation EGFR inhibitor for lung cancer. I mean, at one point in the story, it looked like every major university and every pharmaceutical company was throwing time, money, brain power at this problem, and they didn't stop even after the first generation. So it's really incredible to see, like, there's an example where you have, everyone paddling in the same direction. The global village comes together and they just dedicate themselves to a problem and work at it until it goes away. Um, so, as someone who used to lead research at these organizations, maybe tell us about, how does one marshal these resources? How do you build momentum around an idea? How do you get an organization, and how does a field get excited about an area of research? Because, when it does happen, like, it's pretty amazing.
Yeah, I mean, each of our stories is a great example of what you're talking about. I think one of the quotes I have in the book, which is my own definition of innovation is not only coming up with, something that's novel, but it's showing that it's possible and also convincing others that it's worth doing. So just because you have an idea doesn't mean you are executing on innovation. You got to show that it's possible, right? That's the second part, even with a tool compound, so to speak. But then most importantly, you don't do it in a vacuum. You got to convince others that it's worth doing, which means worth investing in you. Yeah, so I think at all different parts of the value chain, whether you're in academia, pharma, or industry, you got to be able to convince others. I think in the particular instance of EGFR, because the biology was initially, it plays a role in cell growth, and then it may play a role in cancer. And because it was so widely expressed or overexpressed in cancer, people thought that would be a great target for therapy. And then, yes, multiple companies went after it. But even there, one of the illustrations from that chapter is that the companies had an assumption that wild type EGFR was really important.
Hmm.
And so that's what they initially made all their molecules against. But it turned out once you got in the clinic, that wasn't the case, right? And then only 10% of patients who had lung cancer actually responded fantastically, and the rest didn't really benefit. That then spurred an academic question, which is who are those 10%? And that really spurred the academic field to go after that answer. And that's where then multiple groups were working on that, and then we all eventually found the EGF receptor mutations. Now, once that happened, then that sort of unlocked a whole new thing. Oh, there's mutations in lung cancer. That gives a good therapeutic index. Let's make a molecule specifically against mutations that will have an even wider therapeutic index against wild type. And that's eventually how you got to Tagrisso. So I think your question was, how do you convince people? Every step of the way, you have to convince people, but your data may change over time. And then whether it's an academic question or whether it's a therapeutic question or a development question, again, you've got to just make sure you convince others that what you're doing is worthwhile and
Yeah.
it's going to have a big impact.
Yeah. I mean, that's a great example of how important that iteration is required for progress to continue. And, in this case, it's amazing that you got the second and third generation and you went deeper on the biology. And it's sort of interesting how, you get a product and then it raises a new question that goes back to the basic biology and then becomes an engineering problem and becomes a product and then you
Yeah.
iterate and iterate. So that's a great example of when you have the momentum and then the flywheel catches of just what can happen in the field. So we've talked a little bit about
But I
resources.
did want to highlight, even in that one, I then interviewed the AstraZeneca scientists,
Mm,
right, on how they developed osimertinib or what became Tagrisso. And I think what's really fun about that chapter is it sort of highlights even the dilemmas that they face. Like, do we want to just target EGFR T790M? There are multiple mutations, right? Do we just want to target a set of mutations or a broader set of mutations? Do we want to target just EGFR or do we want to target other kinases? Do we want to make a covalent small molecule versus
yeah.
a non-covalent? I think all of us know that almost every day teams are faced with really critical decisions and they can open one door and close another door. And I would just say it's really important that those decisions be made with, the best data possible at the time. But then
Yeah.
people also have to be open to new data that emerge.
Yeah, that is very interesting. I mean, yeah, exactly. These are things that project teams like ruminate over, and then any of those things can take you down a different path because that path is another set of dollars and time, and it's kind of interesting to see where the potential branch points are. Um, so we've talked a little bit about what you can do with resources. Maybe let's talk a little bit about another aspect of this, which is resource allocation. So, is there such a thing as maybe too much momentum and too much resourcing in the field? And I'm thinking about, for instance, the current frenzy of interest in obesity, which everybody's pouring dollars into this right now. And but what about fields then that aren't in vogue at the moment or but are nonetheless still medically important, like rare diseases? So how do you think about, this is maybe getting a little bit philosophical, but how do you think about resource allocation in this case? And um,
Yeah, well, there is this thing called herding, right? And I saw some analysis recently where the herding in big pharma has really gotten greater over the past 10, 20 years. And, that could be a big challenge down the road in terms of then really unlocking, future innovations in multiple different areas. As you mentioned, a lot of people are focused right now on cardiovascular metabolism. Obviously, there's a large potential market there. But the question is how many, GLP-1 agonists are we gonna need or
exactly.
can even make it in the market? Now, I do think, I just talked about this in an interview in Nature Reviews Drug Discovery about China. China developed a number of PD-1 antibodies maybe a decade ago or so. I think there was another motivation at that time, which was the government was investing a lot in Chinese biotechs, and they really wanted to train a whole workforce. And by having them all sort of develop, molecules, it really did train a workforce really fast, and now has positioned Chinese biotech, as very experienced and
Yeah.
knowledgeable and cutting edge in terms of drug development. So that is one potential. But I don't think the herding now, like that we're doing for GLP-1, for example, is has that motive. Um,
Exactly.
yeah, I mean, what could suffer is other indications, but I do believe that in pharma, if you look like every 10 years, new blockbusters arise in different therapeutic areas, and it's really about following the science.
Yeah.
So I do think at a big company, you'd have to take a look at your portfolio. Can you actually compete in GLP-1, or do you just have to say, that's not for us, but we're going to take a different tack, and maybe we won't be the leader in GLP-1 right now, but in 5 years we'll be the leader in something else.
Yeah.
And if history is anything, every decade, as I said, has new blockbusters, and so maybe now is the time to really invest cheaply in other areas for the blockbusters of tomorrow.
Yeah. Yeah, I mean, like sometimes it happens for the good because you're seeing a little bit of a shift back to neuroscience and neurology now, for a while no dollars were going into it. You know, I think
Yeah.
it's also why it's so important to have a rich ecosystem. I think we're very lucky historically, in the U.S. to have a very rich ecosystem of you have, global biopharma, but you also have spec pharma. You also have early startups, and each of these have different folks who are contributing science to it, and also funders, because everybody can then assume different levels of risks and interests, at different stages of development. Um,
I wouldn't discount academia in there. I would say the investment,
I
the investment in basic science also is the ultimate key down the road. So we have to
absolutely.
maintain that. Yeah.
Absolutely, because some of these are less directed questions, but nonetheless, you need to establish that basis of new biology and like what funds basic research, grants and things like that have a different, return horizon than a classic investor or even pharma.
Yeah.
And so, yeah, another reason why the entire ecosystem needs to be continued to be um, maintained. So speaking of resource constraints, let's talk about Hemlibra.
Yeah.
So fascinating molecule, very cool drug, became a blockbuster for Roche, but its success wasn't a foregone conclusion. And in the story you tell, I mean, it seemed like at every turn early on at Chugai, folks wanted to cut funding and cut this program. So tell
Yeah.
us a little bit about this drug and why you decided to write about it, and maybe set
Yeah,
the.
so emicizumab or Hemlibra is a drug that's now approved for hemophilia A. It's a really, really cool molecule in that it's a bispecific antibody where the antibody fragment arms bind to two different things. One binds to factor 9A, one binds to factor 10, and bypasses the need for factor 8, which is missing in patients with hemophilia A. So it really has addressed an unmet medical need and is transforming the lives of patients with that disease. However, the origins of that molecule, I wrote about it for several reasons. One is I did want to highlight a molecule coming particularly out of Asia, and this one comes from Chugai in Japan. And they're generally less recognized in terms of the contributions, and I really thought that was important. Second of all, you'll read in the chapter they actually screened about 44,000 bispecific
Oh yeah.
molecules to come up with this single one. And when I read that paper, I happened to be slightly involved, a little bit involved in the development of that molecule because it came from Chugai to Roche pRed and then ultimately to Roche Genentech. But when I read the Nature Medicine paper about how they screened 44,000 molecules, I was like, oh my God, like this is a real tour de force.
Yeah.
Um, but if you read about it, right, so originally Kuni Hattori was working on actually a molecule that he was trying to not prevent bleeding, but actually clot more to help patients. And that molecule didn't work. Then he was brainstorming with his team. Oh, and one of the team members brought up hemophilia A. He didn't even know that much about it, but then when he dug into it and saw that there was an unmet medical need, and then he conceived of the idea of a bispecific antibody at a time when no bispecifics were approved yet. And
Exactly.
so I can imagine him going to his committee, the portfolio committee, and saying, I'm going to do this. And people are like, yeah, right. But in any case, he did manage to convince his supervisors and funders that he could do this project. And they came up with tool compounds, and then they actually put it into animals. And unlike what they had predicted, they actually got more bleeding as opposed to less bleeding. So
Yes.
you can imagine the team must have been completely devastated. They even had team members, moved from one city to another to take on this project, and then ultimately, the project had to be paused and was ultimately almost killed. But then the team was given 18 months to basically see if they could recover and come up with why, they had caused more bleeding instead of less, and then show that they could get a compound. And they actually went back to the drawing board and looked at the science, and they found that the way that the bispecific antibody fragments were binding actually bound with too much affinity rather than not enough affinity. And so they dialed it down, and they were able to come up ultimately with emicizumab. But it does really highlight, the importance of resilience and persistence, of understanding the science, understanding, potentially why the molecule failed in the first place, in order to then remedy that, and then ultimately as a tour de force in terms of what they did.
Yeah. Yeah, I mean, that is an incredible story. And just, I think, what brought it home to me was, in addition to just being resilience and perseverance, I was really struck by, their motivation, just the attitude and the culture and like the spirit of intellectual curiosity, to go down to the molecular level, to understand what was happening, and really creating an entirely new class before bispecifics had become, common as a tool, was really incredible. So, I just
Yeah,
wanted to share oh, go ahead. Yeah.
no, I was going to say one other element of that is the manufacturing problem, right?
Yeah.
Which I think this molecule really highlights as well. So they came up with the idea of a bispecific antibody, but at that time, first of all, nobody had developed one. But second of all, there's something called this pairing association.
Yeah.
So if you know about manufacturing of antibodies, you have two heavy chains and two light chains, and if you try them to get together, come together in a certain configuration, for manufacturing purposes, you want to get as much of a pure molecule as possible. But if you make a bispecific antibody where the Fab fragments are different, you have this pain chair association problem where your yield will decrease significantly. And so the other big breakthrough of that was done by Tamagawa and others to actually figure out from a manufacturing standpoint, how do I make this molecule and make the yield, greater than 95% in a way that makes the cost of goods reasonable. And so I thought that molecule really illustrated also not only the biological challenge, but then the manufacturing challenge, which is super important in drug development.
Yeah, I mean, it felt like, you needed a stroke of genius at every step along the way. First, we're going to conceive the science fiction notion of a bifunctional antibody. Then the application as a mimetic in hemophilia. We're going to learn how to we're going to learn how to manufacture the drug. Um, that has this paradoxical effect in animal that, where it did the opposite, uncovering that. And then having to overcome, like, your organization saying, you know what, we're going to cut funding pretty soon. I just found it so inspiring that it really wasn't fame or fortune, but just this desire to do the right thing and do it well.
Yeah.
So as a leader, then how do you cultivate that spirit and sort of, how do you sort of maintain that level of just, the curiosity and like, continuing to that drive?
Yeah, I mean, I think that's challenging, but I would go back to this book I read called Zen and the Art of Archery,
Hmm.
which bringing in the Asian background, I guess, in myself, which is, Zen and the Art of Archery is this book about a guy from Europe who went to Japan. He's learning about the culture. He ultimately wanted to learn archery, and so he got this master of archery. And basically, every time he started trying to hit the bullseye, he was only concerned about hitting the bullseye. He didn't care about his position, his form, where his elbow was, where his legs were. And he just kept getting more and more frustrated because he wasn't hitting the bullseye.
Hmm.
But the whole point of the Zen master of archery was like, you got to worry about your form. You got to worry about where your elbow positioning is, where your legs are, where your stance is, what your stamina. And then ultimately you will hit the bullseye. Now, the reason I give that story, and more to me, is like if you're playing tennis, right, and you're only worried about the score and winning, you may not do as well as if you just worry about, am I hitting the ball well? Am I serving to the right corner? And so on and so forth. I think the same thing applies in drug discovery, which is if you're only worried about the end game and getting your molecule approved, you
Yeah.
may get very, very frustrated.
Yeah.
But if every day you are focused on making the best decisions possible, if every day you are focused on doing the right experiments with the right positive controls and negative controls, if every day you're focused on, am I doing an experiment where the negative result will teach me just as much as the positive result, and you love the process, then I think ultimately you will have success in the long run.
Absolutely. Yeah. I think that there is, yeah. And I think the opposite is, also applicable where if that's all you're interested in is getting to the end point, the end is like for something as risky as this, you will always be disappointed. So I think
Yeah.
making sure that there is an art, I think is kind of what I'm hearing you say. There's an artistry to drug development and, wanting to be just excellent in the craft is part of the process.
Yeah, and if you derive enjoyment from being, wanting to be excellent at the craft.
Exactly.
Now,
Yeah.
again, that sounds a little zen, but I do think it will lead to less frustration down the road.
Yeah. I did want to share a quote from your book from Kunihiro Hattori, who was the lead of the Hemlibra product, and it just resonated with me. And he said the quote that you had was, I'm integrating two different parts of your quotes, is He said, I strongly wanted to create an epoch-defining drug. And then later on, when, Hemlibra was successful, I have made my contribution to human society. I'm ready to die at any time. That last bit is a little grim, but I mean, the spirit of that kind of captured, the kind of culture that, the culture of, of innovation that he brought with him. Um, we have some I want to get into some final thoughts, but maybe, before we go there, we're on the subject of, how do how do you get, how do you get resources for an idea that you believe in, but maybe not everyone else does. So any kind of tactical advice you might give, to any sort of the operators and leaders out there about how to sort of get folks to come along the journey with you?
Yeah, I think that comes down to those of us now in biotech. I'm in biotech now, having to pitch
Hmm.
and get
Yeah.
funders, right? Well, one thing I would say is you have to repeat something 7 times until people fully understand. So, just make sure when you're trying to convince others that they fully understand in a clear way what you're trying to do and why you're trying to do it and what's the unmet medical need. Maybe even get feedback as to how it's being received. I think you also have to understand your audience. Are you speaking to deep level scientists who may be super skeptical or even more knowledgeable than you? Are you speaking to people who are more generalist and so on and so forth? So you do have to be able to dial it up and dial it down, at whatever point is relevant. I think from a biotech perspective, you also have to understand the business case. Is there going to be a large unmet medical need? What is the potential commercial opportunity? That's obviously very important, but sometimes can be discounted by scientists. Not because they don't, I don't think because they discount it, but because they're less maybe familiar with that
Yeah.
area. Um, and yeah, and try to ask for feedback if you can get it. I would say the other thing is don't take anything personally.
Yeah.
Uh, you know, I think even in academia, you have to write grants and they get reviewed. And, if you take those rejections personally, you're going to be very, very depressed at the end. So
Yeah.
roll with it, keep going. Many of the best ideas got rejected initially. Actually, even in the book, there's a little anecdote about Lou Cantley, who discovered the PI3 kinase. And
Yeah.
when he discovered PI3 kinase and showed that it was a lipid kinase, there was no such thing as a lipid kinase at the time. And he said he couldn't get funding for like 3 years. And so, if you have the conviction that what you're doing is correct and scientifically sound, keep going, but get feedback on why maybe your story isn't resonating with others.
I think that's one of the great things about the venture community, particularly in our space and how it's evolved, and that there are enough sophisticated investors out there right now, and people who I think still have the spirit of, scientific inquiry and who are willing to take risks, that feedback actually is useful. Of course, there are bad actors out there, but I think when you pitch a lot, there are a lot of smart people. If you can get them to help you think about your pitch, that's a lot of smart people thinking about the same problem that you are.
Yeah, one thing interesting that maybe people don't know who haven't been in big pharma is the question is how do you get so much good feedback with inside pharma? Being now in biotech, you have to pitch to a lot of different people
Yeah,
and to pharma. And every time you pitch, you get great questions and feedback, right? But in pharma, you try to keep your stuff under wraps. And so
yeah.
I've been thinking if I was ever to go back, like how do you create that same kind of environment where the internal teams get also the same level of feedback?
Yeah, in fact, I think in almost every single case, it's always these there's of course like technical serendipity, but there's also relational serendipity almost in every single case where like, oh, this guy was a chemist that was working in a completely different field who happened to be in the same building. I mean, it's just that cross-pollination is so important. Yeah. Maybe moving on to some like last thoughts about where we are today in terms of the overall innovation ecosystem and where biopharma is. I mean, the pace of innovation is just incredible, and it seems to be accelerating. I think that a lot of recent things, remarkably like RNAi, multifunctional biologic, CRISPR, degraders, radiopharmaceuticals, cell and gene therapy, they're all kind
Mm.
of now part of the standard toolkit now. And now AI, a robust Chinese, biopharmaceutical industry and ecosystem are now part of this. So what's your overall take? Hopeful? How do you sort of take advantage of all this, activity that's happening?
I'm super optimistic. I mean, in the very last part of the book, in the epilogue, I talk about how this is the biological century, and really our understanding of biology and then the technologies we have to advance those into therapeutics are unprecedented. So I'm hopeful that we're really going to make a huge impact on multiple diseases. There's still a lot of diseases for which there's unmet medical need.
Yeah.
Last time I checked, cancer is still not cured in the
Yeah.
most most instances, but also Alzheimer's, Parkinson's, still infectious diseases. There's so many autoimmune. And so we really need people to, contribute, to get motivated, to be persistent and resilient, to have what's called emergent learning, meaning learn as you go with the data. But yeah, I'm super hopeful. And, I think also we're going to figure out models where obviously big pharma may be focused on the biggest indications, but I'm also hopeful that in the rare disease instances, and even with ultra rare, that we're going to figure out ways to ultimately get therapies to those patients. I think there have been some movements even on ultra rare gene therapies,
Mm.
where I think in the UK they have a way now that if one vector is approved already in one indication, you can potentially go into another one. We need to come up with really creative ways to cut the cost of development for patients with super rare diseases, but ultimately help them as well.
Yeah. I mean, human biology is endlessly mysterious. There are still a lot of problems to be solved. So I think there's a lot that we could be doing. Um, Maybe a last question before we close then. You've had a unique vantage point for drug discovery, research, and development. So any kind of more general advice or words of encouragement to the operators out there or anybody who has an idea around or conviction around a particular concept? Any words of wisdom to them?
Well, I guess I'm a fan of continuous learning. And so, I think what we do ultimately has to depend on continuous learning because not only is it based on what we've learned in school, but then what we see in front of us every time we do an experiment or once we're in the patients. And so I would just encourage that mindset, growth mindset, obviously.
Yeah,
And then, yeah, enjoy the ride. As I mentioned, Zen and the art of drug development becomes very
exactly.
important to keep yourself sustained in order to make the breakthroughs of tomorrow.
Fantastic. Well, thank you, William, for your time and your words of advice today. And thank you very much for writing this book. So I have a copy of it right here. It's probably showing up in reverse. I hope there's a sequel in the works. And for those of us joining online, there's a paperback version of this available on Amazon now or your local bookstore. I can't recommend it enough. I bought a stack and assigned it as homework for my team. So,
Nice.
and remember to please leave a review as well. So thanks everybody for tuning in today. This conversation was recorded, so if you missed any part of it, you can rewatch it later. And then as kind of a little advertisement, the next virtual event is going to be on March 18th on how to talk to the media, even if you don't want to. So we'll be hearing from reporters sharing their insights on best practices and the issues they're following. And so Elevate will be doing these virtual events year-round, so please share your ideas for future topics that you'd like to hear about at events@elevatebiotech.org. And until then, thanks everyone and have a great day. So thank you again, William. Take care.
Thanks a lot, Jonathan.
Yeah, bye-bye.